Hello, everyone. Thanks for coming along to this Aspirational Neuroscience Journal Club. So we're looking at a slightly different paper today. Looking at this one, Prediction of Neural Activity in Connectome-Constrained Recurrent Networks. So it's more of a computational neuroscience paper as opposed to sort of the engram, physiology, biology, behavioral neuroscience ones that we've typically been looking at, or the ones that we've been doing in the last series, which has been a series of papers on specific model systems where we think a memory decoding challenge prize might be won. So we've gone through things like the zebrafinch HVC RA system, where they store their song memory. We've gone through a bit of Drosophila learning. We've gone through CA3 grid cells. This time we're looking at sort of what could we do if we had a full connectome of a system? What would we be able to get out of it? Now, at the start, I should make clear that my training is more in the behavioral neuroscience, and I'm very much not a computational neuroscientist. So this paper was quite a struggle for me, despite the fact that I found it interesting. And particularly towards the end, there were some bits I definitely didn't fully understand. So if you are a computational neuroscientist and you notice me saying something wrong, or otherwise feel like you want to jump in and clarify something or ask a question about something, please go for it, because you're probably right and I'm probably wrong. But nonetheless, I still found it an interesting paper. So let's get into it. So just a reminder, we're here because the Aspirational Neuroscience Group has this big grand prize for the first demonstration of decoding a non-trivial memory from a static map of synaptic connectivity, so a connectome. And as part of that, we have this one big grand prize for the first group to do that, but also 4 annual research awards, which honor and highlight research into how learning and memory are encoded in the brain. So that's the big picture for what we're doing and what we're gearing up for towards the end of the year, where we give out some of these annual research awards. And this current journal club is a theory paper on, what one could do if you actually had a static connectome, which increasingly we do have these days in the sense of the Flywire data set or other data sets of, for example, Drosophila brains. Okay, so to get into the theory of this paper tackles the forward problem. So often in neuroscience, we have this problem called the inverse problem, which is where we have dynamic recordings from some system, EEG recordings, MRI recordings, intracranial electrodes, some sort of electrophysiological recording. And from these recordings, we are trying to go backwards and infer which neurons were connected to which other neurons. What is the actual structure of the brain from which these recordings come from? Now, unfortunately, this is a famously ill-posed problem because many different connectivities in the brain could be producing these same sorts of dynamics. So it's often the case that it's the recordings themselves don't constrain to a large degree the structure of the brain that these recordings could be coming from. But that's the traditional neuroscience problem that's most commonly studied, because mostly what we have are brain recordings, but not the actual structure of the brain. But in this sort of paper, and increasingly as we have more and more actual connectomes, what we're doing is we have the opposite problem, not the inverse problem, but the forward problem, which is given a map of synaptic connectivity, given a map of brain connectivity, can we determine what the activity that this structure should give rise to is, given particular inputs to that brain? So we're taking the connectome as a given. You can imagine having a C. elegans connectome, a fly connectome, part of a mouse brain connectome. And we're saying, given the structure that we have, can we deterministically know what the activity this structure should give rise to? Or if we can't know exactly, like how close to it can we get for given sorts of inputs? So yeah, in this paper, we're looking at the forward problem. Given a connectome, can we infer the behavior? But this is probably the right point to flag that there's different levels of what one could mean by having the connectome. Where at a minimum, we mean something like you have the wiring diagram, so the synaptic graph of each neuron and how each neuron is connected to each other neuron, presumably plus also the sign, so whether that neuron is an excitatory or an inhibitory neuron. But there's a lot of other things that one could add into this to make it a more complete connectome. So you might have not just these synaptic connections, but also the gap junctions, these electrical connections between certain neurons. You could have things like neuromodulation, so global or semi-global increases or decreases in in excitability due to the presence of things like serotonin or dopamine or other things that are washed through an area. You could have differences in intrinsic excitability. Yes, Randall.
Yeah, just a quick question, because you're listing this under levels of connectome, but neuromodulation, is it meant to be from the connectome, as in you can determine the source of neuromodulation, or is it really just a functional input, something that you've recorded?
I don't have a strict definition here. In the paper, I think where was I talking about? Yeah, I'm not even sure conceptually how to. I would ask you that. Like, how would you characterize if you've got? Well, because I mean, like, it's true that I guess these neuromodulators come from still particular chemical synapses. Like you have dopaminergic synapses or serotonergic synapses or other ones, but
Yeah.
you can have them diffuse out over a slightly larger area. I'm not sure. My point is just like my assumption when I've read other papers is often we're talking about like purely excitatory and inhibitory, like glutamatergic or GABAergic synapses, and that there's more complications in that you can have different kinds of neuromodulation as well, and that can be taken into account too.
Yeah, absolutely.
But I haven't thought through the full conceptual structure.
I don't know. I take back my question because I'm just being pedantic. It's okay.
No, that's okay. That's why we're here.
But wouldn't just labeling? Oh yeah, Zachary, go ahead.
You go ahead, Arsene.
Okay, thank you. Wouldn't just for like what is wrong, for example, if it's just like, let's say, the same way as you have a label for excitatory and inhibitory neurons, just put a label on neuromodulatory neurons and potentially maybe like a small mark of like the diffusion field potentially.
Yeah. There's no problem with that necessarily. I'm just pointing out that you can have the simplest version of a connectome, which is just like a simple wiring diagram with a positive or a negative sign. But we can also get more and more and more complicated in terms of like adding in extra properties. I mean, you can also, I don't talk about it here, but there's also a difference between, for example, modeling each neuron as a single like point rate firing neuron versus modeling each neuron as this complex compartment model, which has its own internal dynamics with complications and different kinds of ways that different dendrites pass signals on to each other, whether those are active or passive. It's just like pointing out that the term connectome is a little bit too loose and that we can imagine different complexities of connectomes, even if we had the full wiring diagram.
Gotcha.
Zachary, did you have a comment?
Yeah, I was just gonna. This is an area that I'm trying to learn more about, but I do know that if you have like a connectome, and often, if we model those connectomes as RNNs or leaky integrating fire neurons, whatever, we usually make this assumption that every synapse in the connectome is like directly driving downstream activity. But if there's a neuromodulatory synapse, and this is where like I need to brush up on my neurobiology, but it seems like then you would maybe want to model that synapse differently, like rather than it having a direct impact on like a postsynaptic potential, like maybe it has like a gain effect or something. And so I just wanted to kind of take this chance to ask, like, because we're talking about wiring diagrams and neuromodulation, but like, do folks know like at those synapses, if it's like a dopaminergic synapse, how we should be modeling that computationally, or is that kind of an open neuroscience question?
It's certainly beyond my knowledge as a non-modeler. Maybe somebody else knows, but otherwise I'm just not sure.
Ken, is there a chance that you might know the answer to that?
Not off the top of my head, but people definitely model like D1, D2 synapses in the basal ganglia. So I'd have to look up how they do it, though.
Boris?
I've seen computational models which do have some spatial structure and do allow exactly what was mentioned already in some surrounding fields and some distance, and then you have some action at the distance with some delay and this type of thing. I mean, it can be modeled. It's not usually done.
Cool. Great. All right, I will keep going then. Yeah, non-spiking. This is just to say that there's a variety of additional properties that you could add into your minimum connectome to make it more and more complicated. Which is also to say that if you have just this sort of simple minimum connectome, that there might be a bunch of biophysical parameters that you've left unconstrained by your model, which is some pretty heavy foreshadowing for what we'll be talking about in a minute. Now, just to point out that I'm not entirely clear on things, the terminology, but one term that I've heard used for talking about not just a simple connectome, but a connectome that also includes things like what kind of the neurotransmitter type used at that synapse and the particular strengths of those synapses and the intrinsic excitability, all those sorts of additional properties I've occasionally heard referred to as a molecularly annotated connectome, as opposed to the kind of connectome we're talking about today. But for now, for the moment, in data sets that are published on, typically what we still see are they get published with many of these parameters still being unknown. So I think of, for example, the Flywire paper that was released in 2024 now, which did not list out a whole bunch of these various parameters that we would be interested in. And when you see the like efforts to model these things, like the Shu et al. Paper, a lot of these parameters were not modeled in those models of the connectome. Okay, that brings us back to this particular paper and what they did, which is they used a system called teacher and student networks. So basically, the setup was they would have some ground truth recurrent neural network that was a stand-in for a circuit with a known connectome. So they build this 300 neuron recurrent neural network. You could record from any of its neurons. Its connections were all known. And that would be the ground truth teacher that was used. And it would be trained to do some particularly simple task where, for example, given different inputs, it would be producing different sort of oscillatory output responses. And then what they would do is they would take a student model. So they would take the teacher, essentially, and they would make a copy of it. They would keep all the same synaptic weights as the teacher, but they would change some of the single neuron parameters. So for each of those 300 neurons in this recurrent neural network, I'll go through it in a second, they had a variety of parameters beyond just the synaptic weights that connected them to each other, and they would scramble those. I see a comment from...
Don't pay too much attention to it. Sorry, I'm just advertising our work.
I see, nice. Feel free to do it. So one of the things I came across, because I wasn't super aware of this like student-teacher setup paradigm stuff, is that in sort of a more classical work, you might have a system where the student is trying to learn the synaptic weights of the teacher, in which case there's like, for n neurons in the network, there's n squared unknowns. There's the connection between each of the neurons that it's trying to learn. Whereas here, there's only n times the number of parameters. So in this case, 2n parameters that the student is trying to learn, which is quite different from this classical student-teacher case. So the particular model they used of each of these single neurons, each of the 300, let's say, is each one is just a rate-firing neuron. So this R of t that takes in the currents that it's received from, which is this x of t. And then there's an additional bias, like an intrinsic bias that particular neuron has. And there's an intrinsic gain that particular neuron has. And these are the parameters, this gain and this bias, that differ from neuron to neuron and that are able to be changed by these the learning processes or the training processes that are going to happen during this paper. So again, they've got the student has exactly the same synaptic connectivity as the teacher, but for each neuron, its gains and its bias may differ. Yeah, I think that is just saying again what I was describing. All right, so let's jump into the figures. Sorry. So at the top left here, we have a diagram of what this system is actually doing. So this is an example of the teacher recurrent neural network. It can have different inputs, different sort of, oh, yep, Ken?
I'm sorry. You showed one equation and it was not a differential equation, but there is a differential equation in the paper where X is a leaky integrating fire, I believe.
Yeah, it's describing the model of how the current changes for the cell over time. From memory, it's got it's that the cell is it takes in its previous current, and I can hold on. I'll pull it up so we can see it. But maybe I can answer your question while I'm pulling it up.
Sorry, the reason why I'm asking is I'm trying to understand at the start what the complexity is of the neurons, and these are not just linear. These are like I was saying, kind of leaky integrating fire.
That's what I thought. Yeah, they're like LIF model neurons. Yep.
Yeah, I mean it's not linear as well insofar as it has this nonlinear function here. Is that psi that takes in the current and the bias and then performs some sort of nonlinear. Let's see if I can remember. Is this a ReLU function? It's where it for like as the current increases. Yeah, Randall.
Yeah, I mean, I know it's important to know what it is in the paper, but I think we shouldn't focus too heavily on exactly what the function is, because the whole point of making this nonlinear model in here is just to have an example of a teacher model and then some student that you're trying to get to approximate what the teacher is. So you can imagine that you could extend this where instead of modeling the neurons as leaky integrating fire, you could be modeling compartments or whatever. It's really the principle that's being investigated here.
And I will also note that later on in the later figures, they will mess around with this nonlinearity and how that affects the particular model, where they both have a model of these neurons as if they were linear functions instead of nonlinear functions. And they also have a model where they change the nonlinearity function between the student and the teacher models and see to what degree that affects the ability for the student to actually learn the teacher's performance. So we are going to see some examples of messing with this as we go on. But yeah, good thing to pull me up on, Ken. And again, anyone else, feel free to pull me up on these things as we go. So as I was saying, this is a model. This is a diagram of how the teacher recurrent neural network works, where for different inputs, it will provide different sort of oscillatory traces. So you can see for input 2 in the light gray, it has this oscillation that dies out earlier, where for input 1 in the black, it has this oscillation that lasts longer in this sort of simulated time period. And that the different neurons of the 300 neurons in this model, some of them are excitatory, some of them are inhibitory, and they have these gains and biases that can vary between, what is that, like -3 and 3 or so. And for the gains between 0 and 4-ish or so, based on the particular input that they're provided. So this is just to give you a flavor of what this actual system looks like. So here's our first sort of results, which is when they are providing a student recurrent neural network, the activity that is generated by these teacher recurrent neural networks, and they're performing gradient descent to train the student to produce the same output given the same input as the teacher by changing those gain and bias parameters. you can see that they're successfully able to learn the task. And this is logarithmic on the y-axis. So they dramatically decrease their error across the training period. So the student networks are definitely able to recapitulate the behavior that the teacher network is doing, which makes sense. They have exactly the same synaptic connectivity. They just got to shift their gains and their bias parameters around until they start producing the same behavior as the teacher does. But what's interesting straight off the bat in figure 1 is that they are not recreating this particular behavior by just shifting their parameters to be exactly the same as the teacher's parameters. So you can see that while the error in their output decreases exponentially, the error in their activity, if anything, increases over time. So when they say error in activity, what they mean is for each given neuron, so let's say neuron 5 out of 300, does the activity of neuron 5 out of 300 in the student network, is that activity the same as neuron 5 out of 300 in the teacher network? And that's taken for each individual neuron from 1 to n in the student network versus the teacher network. And you can see that as the training goes on, the level of error actually increases and then plateaus. And that's compared to this gray line, which you can see here, and which is going to reappear on a lot of these different figures, where the gray line is if you took the teacher network and you just shuffled which neuron was which neuron. So in terms of the labels, so you took 0 to 300 and you just relabeled. Now neuron 5 is neuron 15. Now neuron 100 is neuron 120, et cetera, et cetera. So what we're seeing is, yes, that this error in activity goes up, and that this error in bias goes dramatically up, and the error in gain sort of stays high and doesn't plateau down to basically a 0 level. So the headline from this is the student networks are learning to do the task, but they're not doing it the same way with the same neurons as the teacher networks are. You can see some examples of individual traces for individual inputs down in the bottom here, where you can see the teacher network's in black and the student network is in, well, the various ones are in these various orange flavors. And you can see that they really are doing it for different kinds of inputs. But if you look at some of the traces of individual neurons, you can see that for both excitatory and inhibitory neurons, the trace of an individual neuron from the teacher network in black is very different from the traces of the student networks in orange. It's just to visually inspect this. There's not an analysis to be had beyond that. So I found this pretty interesting just to see this initial example. On the one hand, it's perhaps a trivial finding to show that, if you can have these gains and bias parameters be set differently, that they can, there's multiple ways to perform the same task still. But I found this interesting in that this wasn't really something I'd thought about before in terms of how you could have this happen. So does that make sense before we move on to the next system? Any hands? Yes. Arseni and then Ken. Well, yeah, a bunch of hands. All right. We'll go Arseni, then Ken, then Zach.
Well, I'll guess I'll go with some foundational questions here. So could you please move 3 slides back? Yeah, I guess. Yeah, hold on. This one. Yeah. So didn't quite get. So the 2n unknowns basically means that it's the gain and bias that are unknown, right? Okay.
Correct.
So in terms of, I guess it just comes from my lack of knowledge about how RNNs are trained, and specifically in the student and teacher paradigm, how they are trained. But could you give an example of like one epoch of what is going on there, and in the classic one, right? And how it's different here.
So I will try my best, and someone else again can jump in. So the idea is there's 2 different recurrent neural networks. There's some ground truth teacher, and it has a particular synaptic connectivity matrix, and it also has various parameters for each of its neurons within the recurrent neural network. So let's say for neuron 1 to neuron n, there's neuron 1 is connected to all these other neurons. And also neuron 1 has a particular gain and a particular bias parameter. And then if we feed an input into that teacher network, for each particular input, it produces some particular output. That's all the information that we have. Then for some student network, it might have, depending on what we're trying to teach it, it may or may not have be initialized with the same synaptic weights as the teacher network, and it may or may not be initialized with the same single neuron parameters, so those gains or those biases. In this particular circumstance, we're initializing it with the same synaptic weights. So let's say if in the teacher, neuron 1 is connected to neuron 5, neuron 7, and neuron 100, then the same for the student network. It will also, neuron 1 be connected to 5, 7, and 100. But it doesn't have the same initially, the same parameters for each of its single neurons. So the gains and the biases on neuron 1 will have some random initial value. So rather than being, -5 and 7, it'll be 3 and -2 or something on those parameters. And then what happens during training is that there's an error provided at each particular training time point where the student network at time point 1 will be given some input and it'll generate some output. And if that output doesn't match the output that the teacher would have, then it gets provided an error, and I think it performs some standard backpropagation gradient descent.
Gradient descent, yeah.
And adjust those parameters back to closer to what the teacher is outputting.
All right. Yeah, that makes sense. And then the graph of activity, right? The gray and the black line. What's exactly this? Yeah. So the A figure on the right.
The A figure on the right. So this is when this is a visualization of what the teacher recurrent neural network is doing. So it's given a particular input, so particular currents into each of the neurons. So remember that for each neuron, its like rate firing level was like a function of the current that it was provided plus some bias with some nonlinear function. And then the output of the system, it is true, I've forgotten how they do the full output. I don't know if they're just summing across all of the neurons at the end, or there are particular output neurons that it's recording from. That's a good question that I've forgotten exactly how they defined.
I believe that they're just giving two different input stimuli and then training the network to produce different output stimuli. So I think what you're seeing there is there's input 1 stimulus versus input 2 stimulus, and then the readout that's showing like what the network is supposed to produce in response to those different stimuli. And then you can do like backpropagation to train the parameters to.
Yeah, the behavior of the network is super simple because it's just getting two different kinds of input stimuli and producing one potential output behavior. Yeah.
And I also just want to flag, like, I think something that they've made maybe a little overly confusing in the paper, but that I think is like part of the key result is that like we were talking about, like, oh, like 2n parameters versus n squared. These two model types, they're the same model types and they have the exact same parameters. What they're playing with is what parameters are known and then what are unknown to be trained, right? So the beauty is that those parameter sets align perfectly, but that 2n versus n squared, in one, they're saying we're gonna set the gain and bias to be known and then train the connectivity parameters. That's the n squared. Versus in the other, they're saying we're gonna set the connectivity parameters to be known and then we're gonna train the gain and bias parameters, which are the 2n.
I see.
Yeah, sorry, I didn't phrase that properly. That's what I
interesting.
meant to say, but thank you for clarifying.
I think the paper doesn't call that out, and I think that's like part of the beauty of the experimental setting.
Yeah. It makes sense. I guess the last question on this, so input 1 and input 2, is it one input is into, it is just like examples of like, okay, this current would produce this kind of readout and this will produce that, or that's the input into the teacher and into the student?
I believe it's just different pulses. So
But
Okay,
the
so it's
input
just an
to the
example.
teacher and student has to be the same.
Yeah.
You're
Oh,
testing
I
with the
see.
same input data.
All right. Okay. But the readout is going to be different because the activity. Okay. So, okay, got it.
Yeah.
Thank you so much.
Cool. Ken?
So I'm not getting something about the gray line. So I usually would think that if you scramble identities of neurons, that you would get the worst possible result, but this is actually not the worst. So can you explain the gray line
Yeah.
again?
Yeah, so you're talking about like this gray line in figure D, for example, where it's the
Yeah.
error seen. So critically, the gray line doesn't appear in figure C or these other figures because the gray line is not about scrambling the synaptic connectivity and then looking at the performance based on input and output. The gray line is looking at the error in activity between the ground truth teacher network and some other network with different labels on its neurons and potentially different gains and biases. So what's happening in D is you have the same teacher network that's essentially its structure is exactly the same. So you give it a particular input and it gets given a particular output, and it's doing that just fine. So for all intents and purposes, each of its neuron 1 to neuron 300 has the same gains and bias values for its parameters. But what has happened is in this sort of control example where they're looking at the error in activity, they've just renamed, like shuffled the labels so that now neuron 1 is neuron 100, neuron 2 is neuron 250, whatever. And they're like, what is the error between the original labels for those neurons and the new labels for those neurons, but without actually changing any of the structure of how that system works. But that's when they're not doing any training on that system at all. So they're not then changing the values for those gains or bias parameters. They're just changing the labels around, and they're looking at if you just change those labels without changing any of the actual gain or bias, then what is the error in activity, quote unquote, error in activity that you see between the original system and the now relabeled system?
So,
Does that make sense? Or is
well, it does make sense. I guess it does make sense, except from an interpretation point of view, it's basically saying, okay, I've got neuron 1 and I've got neuron 100, and they're doing their own things in the teacher network. And if they were doing exactly the same thing in the student network, then that would be 0 in terms of error activity. But now,
Yeah.
if I were to swap those, you would think that they would be very, very different. But how I'm interpreting this, you can tell me if I'm interpreting this incorrectly, is that the overall teacher network, the activities don't go that high or that low, and so the difference between them is not that great. But if you let it train the heck out of the network, you can get some just really bizarrely large activities, and that's why the training set becomes higher than that gray line.
Yeah, I don't have a good sense of why the sort of orders of magnitude for why the errors differ between the trained, retrained versions versus just the shuffled version, where what the shuffled version is like 10 to the
In any
one,
case,
like,
I mean, I think the main thing that you're saying is that that gray line is not. I mean, a lot of studies, that type of a shuffle control would be very, 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 very important. But in this case, it's not super important, or at least I'm going to imagine that it's not important.
yeah, I don't think I think it's just a conceptual sense of like if you have the same system with all the same parameters, but all you're doing is changing the labels, how much error is that compared to if it actually does have different biophysical parameters, like different gains and biases.
Okay, thanks.
Yeah, exactly. There are lots of different ways to implement the same behavior, but if all you're doing in the end is just reshuffling the labels, then it's not a big difference.
All right. So that's what happens if all the system has to play with is the inputs and the outputs. That's all it can say. In figure 2, though, they ask a different question, which is if instead of training the student neural network based on the output from the input and outputs from the system, what if instead it gets trained based on being able to see some of the actual electrophysiological recordings from some of the neurons from the teacher recurrent neural network. So this time they're going to try and train the student recurrent neural network based on seeing somewhere between 1 and 250 of the activity of the neurons from the teacher network. So a different sort of paradigm. And we're going to see whether this will produce the same, will train a student to produce the same individual neuron-based activity as the teacher network. So what you can see here is so the labels for the colors are in this M, where M is a proportion of N, where dark blue is only 1 neuron, the yellowy orange is 250 neurons, so 250 out of 300, and you can see it range here. And what you can see in B here is that the error in the recorded activity, so the error of the neurons that have been recorded from, that are the same recordings in those teacher recurrent neural networks versus the student networks, decrease exponentially as the training goes on. Sorry, I saw some questions. Are the weights still fixed? The synaptic weights are still fixed, yes. It's just the gains and the bias parameters that are changing. But for this one, the weights between the teacher network and the student network are the same. Oh, sorry, in on the left here in B, and on the right, we're going to do something different. I'll talk about that in a second. Actually, I'll talk about that now. So yeah, they do have 2 different sort of things that they do in this experiment. So B on the left, they keep the weights fixed, and they allow the gain and the bias to change. On the right, they're keeping, I can't remember whether they're keeping the gains and the bias fixed, but what they're definitely doing is they're allowing the synaptic weights to change on the right-hand side. So it's 2 different versions of what if we keep the synaptic connectivity fixed and what if we don't keep the synaptic connectivity fixed. So that's for ARKit. And then we'll come back to discussing the Lapalainen paper later, because that also comes up in the discussion.
I'm meaning that to be like a side discussion. Sorry.
Yeah. Sorry about that. So yeah, we can see that the behavior, the error of those recorded neurons decreases exponentially as training goes on in both circumstances. But what is quite interesting is that if we look at the unrecorded neurons, so these are the neurons that weren't part of the set that were, the student network had direct access to coming from the teacher, you can see a difference between what happens when only a few neurons are provided and when lots of the neurons are provided from the teacher network. Where when only a couple of the neurons' activities are provided, basically the error in all the other neurons, so the unrecorded neurons, stays high throughout the entire training period. Whereas when more and more of those properties, when more and more of the activity of the teacher network is provided, we see more and more of the error drop away across training, where you can see there's some sort of critical threshold here around like 30 or so of the neurons being provided. Where if you give it enough information about the teacher, then the student network starts to resemble the teacher network strongly. Although that's just for the activity, not necessarily for the actual parameters themselves. So what is fascinating and what they note is that while the activity of the individual neurons starts to resemble that of the teacher, the actual values of the parameters of the connectome-constrained network, their error rate stays high. So it's not like those gains and those biases are becoming exactly the same as that of the teacher network. It's just the activity of the individual neurons is starting to resemble that of the teacher network. In contrast with the training the synaptic weights versus just training these parameters, the errors are just really high throughout the whole thing. It doesn't really learn to recapitulate the same activity as the teacher network. Does that, yeah, Arseni, go for it.
By error gain and error bias, what we mean is literally after a training epoch, we just compare the those two parameters of a neuron in teacher network and student network. That's it.
Yep.
Okay. Yeah, thank you.
All right. Up, Zachary.
Yeah, I just wanted to call out, like, I think that's one of the main interesting findings of the paper is that error in activity space does not necessarily like match error in parameter space, which I think like for whole brain emulation and trying to find like goodness of fit measures is like pretty profoundly consequential if that ends up being like something that's actually happening always.
Yeah. I would agree. That was also, I mean, I felt, as I was saying, a number of these results when I was going through this paper, I was like, oh, now that I've thought about it, it makes a lot of sense that can happen, but it was good to think about the fact that that can happen. Sorry, Mayuri, sorry if I mispronounced your name.
Yeah, I had a quick follow-up on that, just trying to understand what the student RNN is trying to do. So are we trying to reduce the error in the recorded activity using the student RNN, or are we trying to reduce the error on the gains or the bias parameters? What is the aim with the student RNN?
Yeah, that's a good question. We're trying to reduce the error in the activity, not the gains or the bias parameters. So it's trained for, in this circumstance, to the student network for the neurons that it's provided from the teacher network. So whether that's 1 or 20 or 250, it's trying to train those neurons in its network to have the same activity as those in the teacher network. And it's allowed to alter the gains and the bias values for each of its neurons across its full 300 while doing that. But it's not being told to try and set the same gains and bias parameters as the teacher. We're just looking to see whether that is what it does or whether it comes up with different values once the training's been done. And what this is showing is that it can get very good at recreating the same activity, but in doing so, it doesn't necessarily set the parameters to the same values.
I see. Yeah, thank you.
So to jump into the next figure, what they do in this particular circumstance is they start messing around with the kinds of networks that they are looking at. So in this circumstance, they've got networks of different sizes, so made of different numbers of neurons. And they're also got different levels of like underlying complexity of activity. So in this top one, they have a rank 2 connectivity. So that means its activity really only varies across like 2 different dimensions. So you can see that if you do a principal component analysis on this sort of network, that it really, once you get to 2 dimensions, beyond 2 dimensions, you've explained all the variation in activity that you see. As opposed to other more complex networks where they essentially have an arbitrary amount of complexity to them. And as the networks get bigger, the amount of variation that still needs to be explained, you need more and more dimensions to explain the activity that's going on in this particular system. So it's a full rank set of connectivity in its connectivity, in its activity. What they do here is they look at how many, based on the size of the network. So they have networks that are now just not 300 neurons. They're from 100 neurons to 1,000 neurons. And they're also looking at different levels of recording how many of those neurons. So basically, what proportion of the network are they recording? So to pick this one on the top right here, so we've got networks that are between 100 neurons and 1,000 neurons, going from black to orange. And on the x-axis, we're recording between none of the neurons and all of the neurons. And what we want to know on the y-axis is, as we have bigger or smaller networks, and as we're recording fewer or more of those neurons that we're using to train our student network, how big is the error in the activity in the unrecorded neurons? Does it shrink away entirely? Is it small or is it large? And what they're showing is that for systems that are very simple, that have only like variation in activity based on 2 different dimensions or so, then it's pretty much trivially easy to drive the error in the unrecorded activity down very, very small as soon as you have any number of like recordings from those neurons. So only a very small number of neurons is required before that error goes away. Whereas if you have a very complicated system, then you need to have a reasonably large proportion of the neurons recorded from the teacher network before the student network will start to display activity that resembles the teacher network. And you can sort of see this plot here, this inset plot here, where if you look at the network size on the one axis, and you look at the proportion of neurons recorded on the y-axis, you can see that is increasing essentially linearly. So this is a complicated sort of figure, but basically what it's saying is it's not as simple as you always need, let's say like 10% of some network recorded before you can have the individual neurons start to display activity that's the same as the teacher network. It's not about having 10% or 20% or 30%. It's about having a proportion that is relative to how complex the actual underlying network is. And if it's a simple network, you don't need very many neurons. And if it's a really complicated network, you need a lot of those neurons. Randall.
Yeah, just to chime in, I was thinking that it would be nice to mention, okay, what, for example, if we're looking at an actual brain and an actual brain region doing something, what would be similar to, say, this rank 2 connectivity example versus a full rank random connectivity? And I'm just going to throw something out there. I'm going to say like rank 2 connectivity or low rank connectivity. This, for instance, would be something that you could find if you have a kind of neural coding that is similar to population coding. So many of the neurons are actually encoding the same thing. It would be activating in a very similar way. Whereas the full rank connectivity, that is something like maybe what you would find in C. elegans, where each of the neurons is very specialized. I'm just throwing that out there. Let me know if that sounds right.
Yeah, quite possibly. I also think of something like, and this might be totally wrong, but like recording from a like some simple nucleus controlling breathing rate might have like a lot of neurons that are just like increasing or decreasing activity to drive something, as opposed to some complicated bit of cortex. But yeah, I think your example is probably better. Zach?
I just wanted to call out, I read in depth about half the paper and then skimmed the rest right before this, but, and you'll probably get to this, but I think, Randall, they do use real connectomes, like RNNs constrained by real connectomes.
Yes, they do later on in the paper, yeah.
Yeah, and
Yeah.
I'll let you get to it, but I think the interesting relation to what you were just saying.
So I've just realized we've only got a few minutes left, actually, because it's a complicated paper and we've been going through it. So I will basically skip ahead to that now. This is just to your question before, Ken. In this figure, they just talk through what happens, very briefly, what happens if the nonlinearity between the teacher and the student is not exactly the same? So what if you change that nonlinearity to be sort of steeper or shallower? And basically what they show is that it does affect the error, but it doesn't affect it that much. And it's still the main results hold, where you just need more neurons or fewer neurons recorded in order to have the student resemble the teacher. And also they ask what happens if the synaptic connectivity isn't exactly the same between the student and the teacher? What if there's some like level of error, some I can't remember exactly how they quantify it, but the sigma corresponds to either a small amount or a large amount of error. And they basically, again, show the same results where, be that as it may, if there's some error, you still just need to record more neurons and you can start to get the convergence in activity between the teacher network and the student network. But we can come back to that if we have time. But yeah, I wanted to talk about maybe this last figure before we move on to discussion in the remaining time we have, which is in Figure 5, they actually do, they move on from purely this sort of constructed systems, and they look at some actual biological systems. So they go through a Drosophila larvae, a Drosophila head direction system, and a zebrafinch oculomotor system. So in the top here, they're looking at a model of how the Drosophila crawls. So they've got various neurons corresponding to this part of the Drosophila larva's motor system. And they are setting that up as a system. Now I'm trying to remember how exactly they do this, sorry. So they have a recurrent neural network that's got these 178 plus 52, 178 premotor neurons and 52 motor neurons. They have the, I forget if this forward or backward crawling, sorry, I've forgotten where that particular data comes from and how they've initialized it. I think that's in the teacher system, and they're driving the inputs. This is a little embarrassing. I've forgotten exactly how they do this. So my apologies. You'll just have to bear with me and assume that they have some recurrent neural network based on the Drosophila larva that they are able to drive input into somehow. And they're doing the, sorry, Zach, you go for it if you remember.
Yeah, my understanding was there's a known relationship between some of these input neurons and then the output neuron activity associated with forward or backwards crawling. So they're either stimulating the inputs with a forward crawl motor command, or they're stimulating the inputs with like a backward crawl motor command, and then that's what they're calling their A1 and A2 labels.
Yeah, I think that's what I remember them seeing as well. Thank you for clarifying. So then they're doing the same thing with a teacher network and a student network in B, and they're trying to get the student network to resemble the teacher network's activity once again. And again, on the x-axis, we're looking at how many of the neurons were recorded, and on the y-axis, we're looking at the error in activity. This is phrased in terms of correlation. And you can again see that the same headline results hold, which is that as you increase the number of recorded neurons, you see that the error in the unrecorded neurons starts to drop off substantially. And they also do this by looking not just at the activity of the system overall, but they also look at individual neurons. And these traces are a little bit complicated to read unless you've stared at them. But the gray line is the sort of the teacher signal for these two different recorded neurons. So you can see there's one here that ramps up and then drops off. There's another one here that ramps up and then drops off. And these are what these recorded neurons these teacher neurons are looking like. They're sort of one pulses, then the next pulses, then the first pulses, and so on. And you can see that before training in both these systems, the neurons in the student system are not perfectly resembling those in the teacher system. And that remains the case for when there's only a couple of neurons recorded, these two neurons here. But in the system where more neurons are recorded, so when 24 out of the set are recorded, you can see that after training, the traces are starting to resemble that of the teacher system in the student network. So it's essentially just showing that what the results they've been showing in the previous figures have shown can also apply in these more biologically plausible systems. At least this one for the Drosophila crawling system, this one for the head direction system in Drosophila, and this one for the zebrafinch system, the oculomotor integrator in zebrafinch. They do also note in the Drosophila one. Did someone have a comment?
Zebrafish.
Oh, sorry, zebrafish. Yes, there's too many. As Randall was saying, zebrafish and zebrafinch need a renaming. One of them's got to change. They do note something about how in the Drosophila system, it's interesting that this head direction system requires more than just the underlying connectivity. It needs some of these recordings to bump it into working properly in the first place. But we've only just got a few minutes, so I might skip ahead just to the discussion. Basically, here
I wouldn't mind if you went on a little longer, but that's just me.
Well, it depends on how much time. What proportion of people? Give me a thumbs up if you have to leave straight away at the hour, or if we can go for another 10 minutes or so. All right, maybe I'll go quickly, but not blindingly fast then. So yeah, these are just more demonstrations of essentially the same thing that is happening. And then I think this is my last figure. Yeah. So then in the last figure, what they do is they say, okay, can we actually understand what's going on here? Can we explain what's happening with our results? If we make some simplified version of what we are looking at across these experiments. So what they do here is they remove the nonlinearity and they just have a linear recurrent neural network. And they also remove the gain parameter and they just have biases. So it's a sort of simplified recurrent neural network where the only thing that differs between the different neurons is their bias. And they're asking, when we do the training, when we try and get the student network to resemble the teacher network, what is actually happening with these different values on these biases? Like, how are they changing? How are they not changing? What is causing them to change? And what they show is that some of the parameter values get changed very quickly across training. And they refer to those dimensions of the activity which are driving those changes as sort of stiff. But there's also other parameters which barely get affected because changing them doesn't really seem to change the activity of the system very much. And those they refer to as sort of the sloppy axis, where changing those parameters doesn't lead to activity changes, which reduces the error. And so it's kind of sloppy in the space of what needs to be changed to get those bias parameters to actually come to resemble the parameter values of the teacher network. And they show that the degree to which this is the case depends on the complexity of the system. And this is where we get beyond my computational neuroscience level, because I, while staring at this, couldn't really make myself understand what these singular values are exactly. But I can say that from my attempts at trying to understand this, what they end up showing is that based on how complex the underlying activity is of the system, where we're essentially talking about the rank, where before we were talking about rank 2 versus rank n sort of complexity. Here they're spelling out systems with various levels of rank complexity. What they're showing is as you increase the number of neurons you record, and you get up to recording a number of neurons that resembles the rank of that system, you are pushing down the error rates further and further and further. But as you do so, what will happen, and I think it's this C figure that was the key one here, is that the error in the neurons you're recording will stay very low, start very low and stay low. The error in the neurons you are not recording from will drop down and down and down and down and down as you increase the number of neurons you've recorded. So that just means the activity, not the parameter values, but the activity will drop down. But the biases themselves will not drop below a certain level. And that once your system's activity resembles the activity of the teacher, that those bias values just aren't going to change anymore. And they may well be stuck at values that in the student network are not those that resemble the teacher network. But to be honest, I don't think I'm doing a very good job here of explaining because I did not fully understand the maths that was going on here. So Randall?
Yeah, just to quickly admit that I did a little bit the same thing that Zachary did. The very last parts of the paper, I kind of just perused quickly. So I might not have caught this either, but I'm wondering in this graph, so the effect on the biases, is this for the case where when you record from M neurons, but you're selecting the neurons that you are recording pretty much randomly?
Yeah, this is still random. They do have another figure where they...
Okay, great, got it.
Yeah, I'm not even going to talk about it here, but they have another figure where they select non-randomly and show that you can get away with recording from not very few neurons if you're clever about which neurons you select in terms of driving the error down.
Just like based on my skim, and I don't know, maybe there's a follow-up email or something, but my understanding was they linearize the activation function so that they can apply like more kind of linear algebra analyses. And if you think about singular values as like scaling a transformation, so it's like just the scaling factor, I believe what they're saying is that if you find the neurons that correspond somehow to like the largest singular values, that those are the ones that are impacting the transformation most radically. And so if you can record from the, so at a simple level, if you can record from the ones that have that biggest singular value, then you're capturing what is driving these big transformations, which when you do like stochastic gradient descent and you're taking these steps, maybe like I think like smooths out some of the gradients or the convergence to be better, but
okay, so that's why I was asking. So in C, are they using the information about the singular values to pick the neurons to record from or not? Because to me, it looks like this is still the case where they're not using that information to pick those neurons.
I'm pretty sure in C they're not using that information.
They're not using it. Yeah, there's more figures in this. There's like more subfigures in figure 6 where I think then they talk about the selection part.
Yeah,
but yeah, I think this is also one of those areas where like I think a deep read of the methods is needed to really understand it.
Yeah. All right, let's jump into the discussion. So sort of at a high level, what this paper is showing is that a supposedly connectome-constrained network is not necessarily fully constrained, and that networks with the same connectivity and the same output behavior can still have neurons that have different, essentially, values for their parameters. That, you know, with additional constraints from neural recordings or theoretically from other sort of additional data provided, these networks can still be constrained further to resemble the ones from that they're trying to recreate. They show also that these reconstructions, the activity of these networks is somewhat robust to synaptic weight or activation function errors. You'll have to take that as a given because we didn't really have time to go through it. And they show that these results generalize somewhat to real biological connectomes. And lastly, what I was trying and doing a bad job of describing was that the amount of additional recordings required to get this activity of these recreations to resemble that of the original system depends on the underlying complexity of what they are trying to recreate. And just some key sort of assumptions and warnings about what we've been discussing are, they make very clear in their discussion that all of this depends on assuming that the inputs to the network are known. So they're essentially talking about systems that are near the sensory periphery. So think of something in a motor cortex or visual, in a like sensory motor cortex or in visual cortex, rather than something deep inside the cortex where it's very hard to know what the actual inputs to the system are. They make clear that these parameters, these gains and these biases may stand in for a whole bunch of different actual biological processes. You know, you can't just pick a neuron and pick a gain and a bias, and that then ends up fully describing these neurons. And they also talk about how not all systems may parameterize in this clean way. They talk about C. elegans, which doesn't have these nice spiking neurons. It may not work very well when you try and model it with the sort of efforts that they've done in this paper. And then just to, at the end, bring it back to why we're actually here and running these journal clubs, the relevance to memory decoding. What I took away from this, and again, maybe this is trivial, but I thought it's interesting, is that if you have some sort of unannotated connectome that has your wiring diagram but doesn't have these other values, that it may still be difficult to read out from that unannotated connectome what information is inside it in a way that's compatible with memory decoding. So a particular connectivity matrix might be compatible with many different kinds of outputs and therefore behaviors and memories. It might not uniquely determine what that system has been exposed to. And it's relevant to think about this with respect to two of the papers that they discuss, both in this paper and that we have had nominated as prizes for an Aspirational Neuroscience Award, which is the Shu paper, which did show that from just the synaptic connectivity, you could make these accurate predictions about which neurons would be responding in response to optogenetic stimulation, which maybe pushes back against this conclusion that the synaptic connectivity matrix alone isn't enough. And we can also think about it in the context of the Lapalainen paper, where they did start with a synaptic connectivity as known, but then they did a whole bunch of additional training to try and get the performance of their system working in a way that somewhat resembles what was done within this particular paper. And then, yeah, very lastly, my last point is what this paper makes me curious about is they show that with just the wiring diagram that they don't have enough to have the behavior of the student system resemble that of the teacher system, but they can get it to work if they have some equivalence of dynamic recordings. Well, my question would be for like real biological systems, how much more would we need to add before the student network would resemble the teacher? So do we need just the connectivity matrix? What if we also knew the intrinsic excitability of those neurons? Somehow we were able to read that out of maybe the neuron size or the particular ion channels that it had. What if we had some more biophysiological parameters? At what point does the system actually become fixed in a way where a recreation of it will one-for-one resemble that of the network on which it was based? So I'm just throwing these out here. Now, I know we're 10 minutes over time, so if you need to go, then so long. Thank you very much for coming. But we can also stick around and discuss for a little bit because I don't have to rush off straight away. Randall, you're muted, so I can't hear what you're saying.
My apologies. So first of all, the Lapalainen case, of course, is kind of special because they trained their system to a task. And then they did not test any out-of-domain circumstances. So they didn't test anything outside of that task with the task-trained network. So I wouldn't necessarily compare that directly. And the Shu et al. Model is making predictions about neurons that probably will activate in the sequence, in the path, which is different from giving you exactly what the output is going to look like. So the modulation of those cells. So I wouldn't necessarily count that as the same. But I also am super interested in how you can replace directly recording those neurons, the neurons that you need to record from, so the ones that have the biggest impact if you do a PCA on them, how you can replace that with something that you could maybe get out of the connectome or a annotated connectome. And I think there probably is. And I believe that in the paper, there was already there were some hints being made that you can use the connectome to a degree to identify which neurons are going to have a specifically high impact. That's one thing. And then if you had a library as well telling you which neurons respond in a certain way, so if you can identify the type of neuron and then you know something more about its response, I believe that can help you as well. And yeah, I do think overall that, say, systems in which the rank is relatively low because you're using a coding where, say, populations mean something as opposed to, say, C. elegans, are going to be easier to tackle with more of the connectome data and as opposed to recording data. But that's just, I'll let Ken go now.
No, that makes sense. Just to note, the authors do themselves say that about the Shu paper, where their suspicion is because it was this strong excitatory drive, and they were looking at this motor system, that it was more likely to work just straight out of the synaptic connectivity alone, whereas that might not hold for different sorts of parts of a connectome. But sorry, yes, Ken.
If we look at the case that is just varying gain and biases, okay, excitability, whatever you want to call it, in a real neural network, biological neural network, we would be talking about a particular class of neurons that we had identified morphologically or something, or based on connectivity, and they would have a certain range of gains and biases that doesn't represent like 5 orders of magnitude of unknown, I assume. And so I think my main question here is, because I didn't read the paper carefully enough for this, is do they hint at anything that says in a real situation what the actual dynamic range of these unknown parameters might be? And what was the dynamic range of the parameters that they were really exploring here? Is it like 5 orders of magnitude type of thing? I know these are log plots, so
I don't.
You understand what I'm saying?
Yeah, I don't think it was. I don't have a good answer to you, but I remember they show in this figure, assuming that sorry, I've lost the screenshot, but assuming that the figure here describes properly what they have, they look at single neuron parameters that are between.
Which figure?
That's actually based on the. No. I'm perhaps mischaracterizing. They look at the output activity based on. Yeah, no, I'd have to look this up. It's not saying that directly.
More of a comment.
Yeah, no, it's a good question. / comment. I don't know the answer to. Zach?
Yeah, I just wanted to point out 2 things. And Randall, apologies if you said one of these. I had a quick phone call I had to answer. But one is, I'm interested in whether you could use this method that they have for identifying the most impactful neurons to record to also, like we were discussing, if you have like a connectome and you're missing some key parameter of the connectome, whether you might be able to use that like a similar analysis to identify like, okay, of all the uncertain parameters for all the neurons in our connectome, like these 10 are going to be the most impactful drivers based on the kind of singular value decomposition of the matrix or whatever, and like narrow your evolutionary or whatever simulation approach to search those particular parameters and be okay with a lot of errors in the other ones. The other thing I'm really interested in is extending this to plastic, so networks with plasticity. And I find that like a lot of the computational people, I think this is a great paper and I'm glad that they didn't, because it lets you get a lot of like real math. And oftentimes when you start adding plasticity, a lot of, and highly nonlinear things, a lot of our math breaks down. And so I find that often the computational people kind of live in the simplifying assumption world to make the math tractable. But from a more empirical, like data-driven, just running the simulations and doing gradient descent, I'm curious if we might actually like luck out in that, like if a neuron has intrinsic plasticity, right, where it's like gain and bias are being modulated to keep it near a certain like output distribution. That might actually like work to our advantage when doing like student teacher learning. I'm not sure. Maybe it's a complete disaster, but I just wanted to throw that out there as like a interesting whether sometimes when more complexity might actually make something more tractable.
Well, maybe I have a guess at what you mean, but could you make that slightly more concrete in terms of why you think it might be helpful? Because just like there's a truncation in the values that can be had, or
Yeah, so
yeah,
like with something like intrinsic plasticity, the internal parameters of the neurons are being changed, are plastically changed to try to keep the neuron near a certain like output distribution, which I guess like just at an intuitive level means that that neuron's always trying to be near that output distribution. So like if you make a simplifying assumption that like this is the output distribution, maybe like even if you have an error that like propagates a little bit, that the plasticity actually brings it back. Like in other words, like the plasticity mechanism works to help eat up some of your errors and keep bringing it back to the correct distribution.
gotcha.
Yeah, maybe I'll have a more concrete example.
No, that makes sense. Randall, you just go for it, but you're still
Yeah, I think Zachary already answered his own question largely, so I'm not going to dive into that too much. But I think what this does give you is a protocol where, let's say, you have a system like fruit fly, I don't know, some part of the system, and you're getting a fairly good match under some circumstances and not others. This is going to point you to which neurons should you record from Drosophila in general to get a good understanding of what those cells are like and what dynamics you should be incorporating. And then to apply that again in the individual Drosophila case where you have a connectome available. So it helps you to build up that library of understanding of the cells that really matter, that are going to make a big difference. And in that particular system of that animal, be it human or Drosophila, and then to collect enough data about those that you can then find enough hints in your connectome to apply what you've got in your library. So I'm just kind of sketching out here a way that you might be able to collect enough information about the dynamics in general and the connectome specifically to then be able to get closer to what you're trying to approximate for an individual connectome without having to record in that individual all of the neurons that matter.
Yeah, I guess tangentially off that, what I'm left with when I'm thinking about this paper is thinking about systems in which these sorts of problems do and don't matter. So we talked about, and they talk about in the discussion, for example, the Shu paper, where a lack of clarity on these parameters didn't really affect the fact that they were still able to make these predictions about which neurons would respond to optogenetic stimulation. And that makes me think about, for example, if we had a zebra finch HVC RA system where we think there's this synaptic chain mechanism, would that be a system in which the synaptic connectivity alone is sufficiently important that you wouldn't need to have the specific knowledge of the gains and biases or whatever their physiological equivalents are to make strong predictions about what was actually going to happen based on a particular input, as opposed to other candidate systems where intrinsic excitability might matter more. So like maybe from the hippocampus, where I know that is a larger factor at play. It might not end up mattering for the sorts of experiments we want people to do to win these sort of memory decoding prizes, because we're trying to overdetermine the ability to pull information out. But it is, at least I find it interesting to think about.
I do think you've put your finger right on, like, this is the crucial thing when you're trying to find a system where memory decoding can be won based on the static data, which is you're looking for a system where you know that your connectome is giving you enough information. So I think that's really primary for the memory decoding prize. But I really like this paper, and of course I'm biased, but I like this paper because you shouldn't treat it as a paper that is looking at a specific animal or a specific connectome or a particular model of how these neurons work or those neurons work. It's not like this. This is a proof of principle of how you investigate a nonlinear system like this and what you can actually discover from the connectome and what you would need to know about the individual cells involved. And that is incredibly useful, this kind of theoretical exploration of a proof of principle system like this. And I think that this, for example, already makes a really good case against the whole lo-fi idea of just record inputs and outputs and you can solve the system. That is almost discarded right in the first part of the paper. And it does give us some promise for connectome-based work, right? It just says you need to know something about the systems involved. You need to know something about the different regions of the brain and what they do and which cells are involved and how they respond. And if you know enough about those, then you're probably able to get a lot out of the connectome itself to guide you to a solution that works. That's kind of what I'm getting from this paper. So I found it super promising.
Cool. That seems a good point to end on, unless anyone else has any other comments or questions.
Yeah, my only other comment is this paper actually deserves more attention and more time than we've given it right now.
Yeah.
So I'd love to revisit it in some way in some future. I don't know.
I'm
not sure how.
I look forward.
Maybe in the context of our brain emulation challenge thing, maybe.
My other comment. Oh, sorry, go ahead, Ariel.
I was just saying, I look forward to coming to the carbon copies presentation on it, where someone can walk me through it in a bit of detail for the sections I struggle to understand, even with Claude sitting next to me trying to walk me through the steps. Sorry, you go for it, Zach.
I was just going to say, I think that there's also some, I think that they did sometimes a poor job of setting up the problem, and also like the layout of some of their figures is just confusing, where I feel like they're trying to bolster two very different points with like two left-right figures in the same subplot where they're like changing the x-axis. And Ariel, I thought you did a really good job of like walking through it. But Randall, I think for Epistem, I want to maybe give an internal talk on this paper. This is one I've been looking at for a while. And I might make some of my own figures or something. And so maybe if you want,
please invite me. Yeah, because this paper is like right on topic for me. So I'd be super happy to.
I think it needs like a different narrative than the narrative that they wrote in the paper personally.
Yeah.
Cool.
Awesome.
Well, thanks everyone for coming. We'll be back in 2 weeks or so, probably with a more behavior physiology paper, but we will see you all then.
Yeah. Thank you, Ariel. Fantastic job.
Thanks, Ariel.
All right.